back to Creation Myths. I got to make sure everything is working on my end and on YouTube, and then we can go from there. But I got to make sure everything looks okay and sounds okay first. So let's see here. Nothing yet. Nothing yet. All right. Sorry, everyone. Just got to wait a sec, and then we'll see. Okay, I see me. I I hear me. We're good. Okay, everything looks fine on my end. Okay, so we are here with Dave, who is a commenter on a video. I think it was on Rebecca's channel, and we were chatting, and we're going to talk about macroevolution and the mechanisms of macroevolution. Dave, why don't you take a second to introduce yourself? Tell us about your background, and then we will get going.
Yeah, Glenn, I'm a big fan of the channel, actually. And I've got a little bit of high school education in chemistry. And but mostly, I watch a lot of the channels, and there's a constant theme, and it's sort of mocking creation. And so I wanted to come on and talk about evolution, but just at first, just a few problems that have to be overcome before you would even get to evolution. So,
well,
if that's all right.
Hang on. Before we get into that, before we get into that stuff, two things. One, I hear it as well, and people in the chat are saying there's a little bit of feedback. I think your mic is picking up your speakers.
I'm just using the.
So if you have like headphones or anything, that would solve that problem. Yeah, I can definitely hear kind of a little
Maybe
bit.
I'll turn it down.
But you gotta be able to hear us.
How's that? Is that better?
I guess we'll see. That seems better as long as you can still hear me.
Yeah, I'm good.
Okay, then we're good. Okay. Second thing. I need to start before we get into stuff with a question. And so the question is this. All right, here are some of the comments that led to this conversation for everybody to see on screen here. So I'll read these out loud for everybody. Yo, liar, I'm right here waiting for the invite to embarrass you. You ran your mouth saying I was just a keyboard warrior. Don't trip on your skirt running away. Oh, that's right, you don't know what the definition of a woman is. You continued, I know you are coming here running your mouth to get attention for your channel. Would you like for me to embarrass you here or back on your channel? I watched Kent Hovind embarrass you by asking you for mechanisms that create new body forms. You argued about definitions, then ran by saying you had to get off the call. I can do what Hovind didn't, but explain the science, clown. You continued, everyone knows you are a liar. Like the time you made a video claiming there is observed single-cell organisms evolving into multicellular. Don't run from me, I will come on and embarrass you, liar. What happened? Once you realize I definitely will do a video and force you to answer questions you know you can't, you suddenly stopped all of your pompous nonsense. It will only take 10 minutes. You will be stuttering, then get mad and start arguing about other things to avoid getting exposed as a liar. What happened to you giving me the ability to embarrass myself, liar? So those are some of the comments that led up to this conversation. So the question I want to start with is very simple. Are we going to be able to have a mature conversation tonight, or are you going to keep acting like a petulant 12-year-old? Let's start there.
The comments were only to get me on here. Mm-hmm. So I'm fine.
Good.
We can have conversations.
That's great.
That works.
That's great. Let's have a mature conversation then.
Perfect.
So as we said, let me put us back like this. As we said, the topic that you asked to talk about was macroevolution and the mechanisms of micro and macroevolution. So I want to start here just to make sure everybody's on the same page with the topic of macroevolution. What is the definition of macroevolution that we're talking about?
Let me get yours.
No, I'm asking you because you're saying it can't happen. So I want to know what you mean when you say macroevolution can't happen.
Changing body form.
Changing body forms. Okay. That's not the definition of macroevolution. That word has a meaning, and that's not the meaning, right?
What does it mean anything?
Macroevolution, which, to be
Yes.
clear, again, the whole point of this is you're saying you want to talk about how it can't happen and why the mechanisms don't work. The definition of macroevolution is not change in body forms, to be clear.
The accepted definition of evolution is just it's a change in the allele over time in a
Right.
population,
Yep,
right?
that's evolution.
So where in that sentence is macroevolution? Where does it say anything
Macroevolution.
about creating anything new?
No, it doesn't. Because
So
that's
exactly,
evolution
that's
is defined as the changes in allele frequencies in
right.
populations over time. Yes.
Yes.
Macroevolution is a subset of that is defined as evolution above the species level. So processes like speciation are macroevolution.
Species. Let me stop you there with that one. Speciation. Okay. The most common one we hear about is Darwin's finches, right?
No, there's a million examples of speciation,
The most
but
common one that most people hear about is Darwin's
nope,
finches.
I'm going to stop you right there because I want to get us all on the
Why are
same
you arguing
page.
about this? I'm getting into it.
Because I'm making a point of this because you're running your mouth online without knowing what you're talking
I'm
about. So we're going
I'm
to be
giving
clear
you
about this.
I'm giving you an
So,
example
nope.
of speciation.
Now, here's what we're doing here. I'm telling you the definition of the topic that you wanted to talk about, which is macroevolution, which is speciation or higher. We can go from there, but we're not going to have this conversation about a fake made up definition. Please continue.
Okay, let me just point this out. Darwin's finches, they had a point mutation on an ALX1 gene. It deformed their beaks and created thicker beaks, which were beneficial in a drought, right?
Yeah,
So
but
it's
speciation didn't
basically,
occur in that example.
it didn't occur.
No.
No, I've read papers, and it did.
You're
How did,
talking about the 1970s drought?
no,
No, which drought are you talking about
no,
then?
this was from DNA sequencing.
No,
They
but
discovered.
which, there's the famous drought that with the finches and the beak size is from the
So,
1970s.
so are
Is
you
that the one you're
saying,
talking about or a
are
different
you
one?
saying, yes, are you
Okay.
saying that Darwin's finches did not get thicker beaks?
No, they got thicker beaks. That's
Okay.
not an example of
And I'm
speciation.
telling you, the reason why they got the thicker beaks was a point mutation creating a
Great.
birth defect.
Why do you think that's relevant to macroevolution?
I'm getting to it. If you'd stop
Okay.
interrupting me, please. Okay. So it was a point mutation, created a birth defect, which is a thicker beak. It was beneficial. The whole population took on the feature. The allele for normal beaks was lost. Okay.
No, it wasn't.
That's why the whole population had the same beak. And that's how they became a species, is because that population all became the same with the thicker beaks. And that's how it became a new species.
So
Are
in
you
the
saying,
1970s in the drought, when the beaks got bigger, you're using that as like the example of speciation.
I'm using an exact example of a point mutation creating a birth defect, which created a new species?
And how are you defining species?
I'm not defining species.
Oh,
It's the evolutionists that said it's a new species,
okay.
right? Everybody knows that.
No,
And
no,
so
no, that's wrong. Here's the
the
thing, that's
mechanism
wrong.
that,
You're conflating two different things. You're
no, I'm
conflating
not.
two different things happening. One, there was the drought in the 1970s, very well documented, where the beaks got bigger, but then they actually didn't stay big. They went back to being smaller after a few years of typical weather. There was no long-term change in the beak size. You can see the graph go up and then it goes back down, right?
So you're
So what
saying
you're describing
there
is
was no new
not
species.
what you're describing is not a speciation event.
So when you look in a textbook and you see Darwin's finches, and you see one picture, and then you see the thicker beaks, and you see in there it says new species became Darwin's finches because of thicker beaks.
You are confusing the initial adaptive radiation that occurred on the islands with the
I'm
specific
not talking about
1970s event?
I'm not talking about adaptive radiation. I'm just talking about the mechanism. The mechanism was a point mutation that created a birth defect,
And
which
how are you
changed
and how are you defining it as a birth defect specifically? What makes it a birth
birth
defect
defect.
compared
Birth
to a
defect
new
is
beneficial trait?
birth defect is simply when a body part or a system is born different than the normal. It's not a negative and it's not a positive.
So all of evolution is birth defects.
Birth defect does not have a meaning as positive or negative. It just means it's an abnormal body part or system. That's it.
Okay. So you're
Right?
describing evolution of the finch beak morphology, and this is supposed to be a problem for evolution?
I'm saying that that's the way speciation happened in that situation.
So
A
again, in the
birth defect.
1970s, that wasn't a speciation event, to be clear.
So when did it happen with the beaks?
With the
When did it
finches,
happen with
when they
what
colonized
happens?
the islands initially,
Yeah. So what was the change in the birds that made them a new species?
when they started using different food sources, different populations experience different selective pressures. So you have variation in beak size. Different
Exactly.
populations experience directional selection for different beak
Yep.
morphologies. And over
Change.
time, due to geographic and habitat isolation, they become unable to interbreed with each other. And at that point, they're separate species.
That's
Like,
not true.
Okay, you're
So
welcome
anyway,
to you're welcome to say
anyway.
nuh-uh, but like that's the process. And we
You
are
just
directly
said
observing this. We're directly
just
observing this in a species of flies called apple maggot flies. Like right now, we're using modern genomics to document this
I
process.
don't
out
want to hear
here.
about the apples.
Oh, good. You know the example. Good.
Can we just get to where I'm going with this?
Well, you wanted to talk about macroevolution, so we're talking about
I'm getting ready
macroevolution.
to. Okay. I'm getting ready to. Okay. The easiest way, and I know that when the evolutionist wants to say we're going to, like a body part is built, a new body part is
Okay.
built, they adapt original, existing things, they extend out
Right. Something
genes,
changes. Yeah.
yeah, they take existing genes, mutate them, and build on top of that, right? So let's simplify it, okay? And this had to have happened, right? So let's go to a first simple cell.
Oh,
It
so
would
now
only-
we're going back now.
No,
Got it. We're going to end
get
up
to
with
my point
origins or
here.
bust.
We're gonna have
Everybody
to,
in
we're
the
gonna
chat,
get
you
to
see where this is going?
mechanisms. Yeah. First simple living cell. Okay. It's only going to have genes for basic metabolism and replication. That's it.
Okay. Sure.
So how does it build the first body part?
So you evolve multicellularity first because
No.
you have
You don't
single
have-
cells, and then you have colonialism, and then you have multicellularity, and then you have morphological specialization within that multicellular structure. You don't go from one cell to like a body. That would be silly.
I didn't say that. I said a cell,
Yeah.
right?
Uh-huh.
Group of cells that can replicate. It
Okay.
only has metabolism and replication. That's all it has.
Okay, so
Now,
we're talking
it's-
like prokaryotes,
Exactly.
like original
Mitosis.
bacteria that exist. Yeah.
Mitosis,
Okay.
bacteria, first
Nope.
simple cell.
Nope, bacteria don't do mitosis. Come on, man.
What do they do?
Binary fission. It's a different process. Mitosis
Whatever.
requires the nucleus.
Whatever. Binary fission.
Like, come on. Evolution is fake.
See,
but
you just
don't
wanted to
know
argue about
words.
nonsense. Now, let's get to the point.
Oh yeah.
Okay,
Hang on.
let's
I
get
just
to
want to
the
be clear.
point.
Hang on one
Okay.
second, Glenn, because hang on. So I just want to be clear for everybody. So the first cells were prokaryotes, no nucleus. Mitosis is a process that requires a nucleus. Glenn here thinks you can't get from the first cell to everything that exists today because macroevolution is impossible, but also he doesn't know the difference between binary fission and mitosis. So that's where we're at.
Okay. First simple cell. How does it change or add anything new? It has to have genes.
Yeah,
Right.
there's lots of ways you get new genes.
Okay, be specific. How if you can't mutate the existing,
You can
because
mutate
that
them.
would kill it.
Nope. That's not true.
Yes, you can't take the existing genes that every one of them are required for life and metabolism, right? They have to
Can't
stay the
mutate
same.
them.
They have to maintain their function, right?
Is it gene duplication a mutation?
We're getting to that.
Is it a yes or no?
We- We're getting to that.
The answer is yes, everybody. A gene duplication is a type of
Can
mutation.
you let me finish, please?
I,
I'm getting to that. Okay. So you can't take an existing body part and adapt it because there are none.
prokaryotes don't have body parts. They're single
Just stop.
cells.
You got a cell body, and I'm using an example like, say, a flagella. Anything new that requires genes.
Okay.
Right? So that simple cell, it only has the basic genes. Okay? It doesn't have anything that you can modify. There are no other parts that could be modified.
Well, that's not true, though.
Where,
You can always modify things. Most mutations. Is it your contention that most mutations are lethal?
You're wanting to skip over what I'm saying.
Well, no, I want to clarify what you're saying because you're saying
I'm
you can't
being very
change things
clear.
without dying.
Simple
Is
cell.
it your contention that most mutations are lethal? Or all mutations are lethal? That would
I'm
be
not
silly.
saying anything about mutations yet.
But you said if you change anything, it dies.
If
you can't
if you change a gene which is specifically creating a protein that's needed for life, right? And you change it and it no longer makes that protein, then the cell dies.
That's a lot of qualifications. There's a lot of mutations that change it without doing that.
You can modify it a little, but I'm talking about you're getting from a basic function, right? And then you want to add a body part, like a flagellum.
That's not a body part. Like,
Cell
I don't know
body.
why we're talking about unicellular protein. Like,
You know what I'm talking about.
then
Stop.
I ask you to
Just
use the
stop,
right words
please.
for things.
A cell body and a part.
Yeah, a component of the cell.
That's
Sure.
perfectly
Structure.
clear. That's perfectly clear. Structure, body part. Cell body, body part. Okay. Now to get to that body part, let's just say it's 3 proteins. Okay.
Yeah, can you duplicate genes? Is that a thing that can happen?
I'm getting to that.
Yeah, just yes or no. Is that a
Yes,
thing that can
yes.
happen?
Plants
Okay,
do
great.
it all the plants
Cool.
do it all the time to express protein. I know this.
Great.
So you just let me get to what I'm saying.
No, I'm
okay?
well, it's a conversation. It's not a soliloquy. So
It's hard for me. It's hard for me to explain it when I'm get interrupted.
It's
Just give me a
also hard,
second.
well, it's hard to understand what you're saying when you use the wrong words for things. So I have to like clarify to make sure I know what you're talking
I'm
about.
just trying to keep it simple. Okay.
Yeah. Hey, by the way,
This is
hey, Glenn. In
Yeah.
those comments I showed at the beginning, you said you only needed 10 minutes to embarrass me.
that's because you keep
How's that
interrupting
going?
me.
How are we doing on that?
If you'd let me just do this one example and then you answer the question, we're done.
I would love to hear your example, but you keep
Just
saying things that
let
are
me run through
extremely
it.
wrong.
Let me run through it without you criticizing a definition.
No, I'm going to criticize definitions when you get them wrong. You got to understand
whatever.
that. Like,
we
you
go.
keep going, but
Here we go. Basic cell. You only have the genes for metabolism and replication. If you're going to create something
That's not
like
true,
if you're going to create something like a flagellum, you need genes to make the proteins.
yeah.
Now let's just say
And you use gene duplications from metabolic proteins to
get
do
to
that
that.
because the core of the flagellum is the same as
So,
an ATP synthase. It's the
let's
same.
say, to keep it simple, those 3 proteins, there's just 3 proteins needed. One of them is 125 amino acids, one of them is 347, and one of them is 455. So
Are
the
these
gene
real numbers
duplication-
or made up numbers? Just to be clear.
It's just made up.
Okay, these
this
are
is
made up
just
numbers.
an example.
Okay, it's a
It's
made up
just an
example. Okay.
So the gene duplication is the only way that you can get the genes
That's not true.
from this simple cell
No,
because
that's not true.
there are no whatever. How are
What do you
you going
mean,
to get the
whatever?
genes?
That's not true because you have other mechanisms.
Okay,
Like
how are you going to get the? Okay, you
de novo
you need
gene
the
origin,
free-
horizontal gene transfer,
There are
plasmids,
there are no-
transduction. Like, I just gave you 4 more mechanisms off the top of my head.
Horizontal gene transfer requires genes that are different to transfer. These all have the same thing.
No, it doesn't require genes that are different. It can just be an extra
To do
copy of a
to
different
make
allele.
a change, it requires something different.
Just be an extra copy, and then you
Anyway,
have more of the protein.
okay,
That's
sure.
a difference.
Okay, fine.
Like,
Well, you're not letting me get to the part because you know I'm getting ready to get to the parts can answer.
I can't wait to see how you're going to embarrass me
So,
after all
three
of this.
three
I'm
proteins,
edge of my seat.
hundreds of amino acids. Okay, you have to find some random process that finds those three exact genes that make those three exact proteins. So you've got to you've in a human body.
Wrong.
You've got amino acids anywhere from 50 to 28,000 amino acids. So first your duplication has to find the correct
You're
number
wrong
of
because
amino
you're
acids.
assuming only a single amino acid sequence can do a specific job, and we've directly documented that vastly different amino acid sequences can accomplish the same function. So, like, your assumption right off the bat is wrong.
I just said one's 357, one's there's a length.
Yeah, but you're assuming
Right.
you need those specific 357 amino acids, and that's wrong. You can have a different combination of amino acids that do the same job. I can give you examples of that if you want.
Just
where
let
you
me
have
let me say
different
this.
proteins
Let
that
me
do
say
the
this.
same function.
Let me say this. You've got anywhere from 50 amino acids to thousands of possible combinations of lengths of amino acids. Within every one of those, there's 50 to the 20 to the power 50, 20 to the power 51. 20
Yeah,
to
you're doing the
power
Douglas
52.
Axe thing. Yeah, that's wrong because it assumes a specific target, and that's not how biology works.
There's
You're-
if
there are
the
multiple-
system is random,
It's
then
not.
you can't. It's not random.
No.
Random
Evolution in biology is
mutation?
not random. Components of it are
Random
random.
mutation.
Mutations are random. Evolution is not.
That's the only way you can get your genes.
That's not true. We've been through this. Gene duplication, horizontal gene transfer, plasmids, transduction. There are other ways you can get things than just single point mutations.
Gene duplication still requires you to get the right length and amino acid.
You're again assuming
Amino acid
No, we're
and-
gonna hang on.
and
We're gonna time
configuration.
out. We're gonna stick to this point because you've made this error several times now. You're assuming that in order to accomplish a specific biochemical function, there is one and only one protein that can accomplish that function. This is from Douglas Axe's 2004 paper. That's wrong. We've directly documented that you can have proteins that are very different from each other that biochemically do the same thing. Which means all this math about, X number of amino acids to whatever power, none of that is valid because you can have multiple proteins that do a certain job. And even better, we've experimentally done this where you make random polypeptides, and it turns out some of those random polypeptides accomplish biological functions that actually increase the fitness of the cells in which they exist. So we find new sequences to do certain jobs that don't exist in nature just by shotgunning a bunch of random sequences and seeing what they do. So the underlying assumption of the point you're making that you need to get this sequence, need to get this sequence, need to get that sequence, that's wrong. Just like empirically, experimentally, that is wrong.
See, your papers aren't talking about structures. I'm talking about building a structure, not an enzyme, not a metabolic process, a structure.
When you say a structure, do you mean
Like
like
I said flagellum.
What's a flagellum made of? What's a flagellum made of?
Proteins.
Proteins. So the studies that I just mentioned that talk about random polypeptides,
Yeah, none of your.
What's
none
another
of your,
word for
I
those?
read your paper. None of your papers deal with proteins.
what's another word for polypeptide?
Polypeptides just means more than one peptide.
What's another in a cell? What's another word for polypeptide? Because if I randomly generate polypeptides and they have biological functions, what's another word for those molecules?
They can be enzymes.
And what type of molecule? There's four types. Okay, it's like Bio 1. There are four types of biological macromolecules. There's carbs, proteins, nucleic acids, and lipids. Which one of those categories do enzymes fall into? They're proteins.
I just said that.
Okay, great. So what's the problem then?
Enzymes are like
Structures.
50.
Proteins.
enzymes are not structures. I'm talking about a flagellum which forms like a tail and
Yeah, that's a
spins.
polypeptide. It's the same fundamental structure. It's just a different sequence of amino acids. It's the same
Okay.
biochemically. It's a
So
polypeptide.
so
Like,
you're saying that when you look at the structure of a flagellum, then there's any random number of things that can fit in there to work together. Is that what you're
there's not
saying?
an infinite number, but there are multiple different things that can work. We have flagella that
Okay.
are like missing parts and they still work
Yes.
fine. So the famous example is the E. coli flagellum that Michael Behe incorrectly says is irreducible. There are other types of prokaryotic flagella that work the same way, but they have different parts. They use different proteins, and they're actually
But
missing parts that the E. coli flagellum has. Now, in the E. coli, if you remove those parts, it doesn't work. But in other bacteria, you don't need those other parts. They're extraneous. So, yes, you
that's
can
not
have
even
multiple different structures doing the same job. So, again, I don't know what the
that's
problem is.
the problem is that all of those proteins you're talking about, they have a specific structure, amino acids, and configuration. Now, they may be different proteins in different organisms. For the flagellum, that's fine.
Can more than one protein do a specific job? Yes or no? The answer
They
is
have to have
yes.
an exact.
The answer is yes. For
Yes,
example,
but not in a flagellum.
uh,
If one piece is required for another piece, you're saying you're
there are
saying to
different
me.
types of flagella
No, you're saying
that
to
use
me
different
that
proteins that are non-homologous with each other. Did you know that? There are different types of flagella that use different proteins that are
we're
different
talking
from each
about
other.
one type of flagellum, right? Not anybody, not anything else. One type. It's got three parts. Are you saying that any number of proteins can work together in that system?
No, I've said repeatedly that it's not the case that an infinite number of proteins can do it, but it is more than one, which is what you're saying.
Okay, how many?
A lot. There's a lot of different
Yes.
types of
Okay.
prokaryotic flagella.
No, we're just talking about the one.
Why are we only
So
talking about the one? If the
in
problem is
in the
no, listen,
ones,
if the problem is that such a structure can't evolve, you have to consider the full universe of possible structures that accomplish that function, right? You can't just say, "Oh, this one thing, okay, there's 19 more." So
you
it's not
still
just about the one that you want to talk about, it's the function of a flagella that uses a proton-driven rotor, right, that
right.
spins in order to accomplish locomotion in a prokaryotic cell. There are many ways of accomplishing that.
I'm not talking
That is
about accomplishing how it spins. I'm saying, how are you going to find those? How are you
how are you
going to
going to find the
generate?
proteins? Because there's a lot that will do it. That's what I'm saying. So it's not
You have
that you need this one specific combination of amino acids will do it. This is the fallacy that creationists have drilled into your brain, and they're wrong about it. And I want you to just think for a second about how you've been lied to here. Okay? Because again, this goes back to that Douglas Axe paper from 2004, and creationists have run with that for the 2 decades since. And the fact of the matter is, they're lying to you because we have direct experiments that show you can get the same function from multiple different proteins. I'll give you another example just to keep it rolling. I'll give you another example. In HIV, you have to inactivate a human protein called tetherin. It's a part of our immune system. It prevents the viruses from leaving the cell after they infect it. Now, one type of HIV, you've
four
got a
amino
protein.
acid substitution.
Yeah, exactly. You've got a protein called VPU that evolved
Yeah,
three new protein binding sites
yeah.
with
It
between
was a.
4 and
Let me.
7, right?
It's the same time. Four
There's
amino acid substitution in a highly
I'm glad
mutated
you
way.
know this irreducibly complex example that we observed
Yeah.
evolving.
So it's
Great.
about a
Did
one
you
in
know
160,000 chance.
Now, did you know that an HIV, so that's HIV-1 group M, and
That's
HIV-1
simple.
group N, the virus accomplishes the same function, but it does it through a different way. The amino acids that do it are completely different. But it's another set, and that one actually has more mutations. It's like 10 or 12. And so that one has a
See,
completely set of amino acids that accomplish the
you're
exact same function.
you're talking about existing things that are just altering
That one
themselves a
didn't
little.
exist. That one didn't
I'm
exist.
talking about
That's
how
a
are you
new
going to create.
function. Hang on. No. Don't talk over me. That's a new function that didn't exist prior to HIV jumping into humans. Like, that's a new thing, to be clear.
Okay. Let's say there are 100,000 proteins, different proteins that can do those three things. 100,000. Okay.
That's probably high, but sure, whatever you want to
Whatever.
do. I don't know why you're harping on three made-up proteins, but sure, knock
Because
yourself out.
I'm trying to get you to the point where you've got to come up with a macroevolution mechanism that can create millions of proteins by some random process to take a simple cell into all life forms.
Okay, but the thing that
And
you just described, the creating all the proteins, that's not macroevolution. Macroevolution is speciation or higher. And
that's speciation. That's
you
higher.
don't need new genes to do that.
Oh, really? You have the same genes as a one single cell?
Nope, you don't need new genes for speciation to occur.
I never said that. It's a mutation.
Is a mutation the same thing as making a new gene?
You're
No, it's
avoiding
not.
you're avoiding what I'm saying.
I'm having trouble understanding what you're saying, if I'm being honest.
I'll be very
with you.
clear. A first simple cell
Maybe someone in chat could explain it to me.
must somehow evolve millions of different proteins by some random mechanism
Nope. Not all
to,
evolution is
to
random.
accomplish all life.
Not all
If it's
evolution is
not
random,
random,
so
it's
that's wrong.
guided. If it's not random, it's guided.
Nope.
Which is it?
Nope. You can have weighted probabilistic processes. You can have selective processes. None of those are
But the
guided.
selected only comes from a random thing that's selected for. So the random thing creates the thing that is selected for. That's not new
So
information.
you're saying mutation, right? You
No,
need to have
I'm not saying
mutations to
mutation.
generate variation, and then those variants are selected for, and that leads to speciation. Yes?
I didn't say anything like that. I said a single cell must create
Chad, help me out here,
millions
man.
of
I
new
can't
genes and
translate.
proteins to evolve into all life. I'm
Yeah.
asking, what is that specific mechanism that creates all of
I
these new genes
gave
and
you
mechanisms that work
5.
together?
I gave you 5 before.
You didn't give me a single one that builds a new gene.
I literally did. De novo
Name
gene
one and then we'll
synthesis.
go through it.
De novo gene synthesis builds new genes. And like
Yes,
this is
but
a very
how does it create new proteins?
what does a gene do? If
No,
it
I'm
makes
saying
new
if
genes, what does a gene do?
The de novo gene must then change. It must have changed from something, and now
Right.
it creates a new useful
Right?
protein.
It was a
There
non-protein
is-
coding region, and then it became protein coding. Yes, that's a new gene that makes a new protein.
Yeah,
Yes.
and I'm saying
That's part of the process,
That is
yes.
that's the mechanism that must happen millions of times.
And it has happened a lot. Also,
You
you don't
can't
have to
name a
have that
single
happen
time.
millions of times because you
There's-
have
there's-
gene families. So each gene family has an independent origin, but then within each gene family, you have a bunch of related sequences from gene and genome duplication. So you keep trying to say it has to be this process, or it has to be that process, or it's all of these processes are happening simultaneously. Like, I don't know what the problem is that you're trying to articulate. There are lots of ways we can get new genes. So you can have a gene duplication, and then one of those copies becomes something different. That's a new gene. You can also have a region that previously didn't code for a protein, becomes translationally competent, and now it codes for a protein. That's a different way to get a new gene.
I could care less about genes. It's proteins.
What do genes make?
They have to make the correct protein.
They make proteins.
Not just
so
not a gene that becomes just duplicated. It must somehow change to create the new useful protein.
yeah.
So it must somehow change the number of amino acids it makes
Not
and the
necessarily.
configuration. And if it doesn't change, then every single gene makes the same amount of amino acids in their protein.
No.
So it changes.
Of course it changes. It doesn't
And
have to.
it
but
must
can.
find the right number.
What do you mean find the right number? Like,
Number of
it's
amino
not
acids.
evolution. Do you think evolution is like looking for specific proteins? Like, what? What is your
I'm saying it's not
understanding?
looking for anything. I'm saying it's random. It's all random.
It's not all, again, evolution is not all random because say you have a gene duplication, that is a process that tends to happen at recombination sites. The genes adjacent to recombination sites are where the duplications tend to happen. Now, those duplication sites are not deterministic, but they're also unequally distributed. So it's like a weighted die, right? It's not always going to come up as 6, but it's more likely to come up in certain places than others, right?
But it's still
Now,
random.
it's probabilistic or stochastic
It's
or better
still
used than
random.
random.
Still
Now,
random.
when you have a duplication, one of those copies will often mutate. And when it mutates, it biochemically does a different thing. Now it's something new. Now it's
But
doing a new function.
but it has to be useful.
And
And
many times it is.
that
And
means
many times it is.
out of an infinite number of possibilities, it has to pick the right one
No, it doesn't have to pick the right
to
one.
be
It has
useful.
to pick something out of the enormous universe of beneficial
Yes.
things that can
And
happen.
if you're trying to build a system, then you need the right proteins to be found.
You know, we have like directly observed de novo gene origination, right? Like the
I'm not
process
saying
that
there's
you're
not.
getting hung up on. We know we've
I would like
observed
you
this
to hear
happening.
de novo protein.
De
My
novo
genes.
gene origination is the same thing as de novo protein origins because
No, it's
genes
not.
in the genome code for proteins. This is
A
like,
de novo, a
this
de
is
novo
Kent
gene is
Hovind
just
level.
a
Like, are
gene
you
made from
telling
scratch.
me, say that again?
A de novo gene is just a new gene made from scratch.
And what does the gene make? The gene makes a new protein. So in order to get new
Now,
proteins, you have to get new
now
genes, right?
the de novo gene
Makes a new
must make
protein.
a useful de novo protein. Useful. Not de novo protein, but useful. So it has to be able to build a system of
It
new
has to be able to
proteins.
interact in some way that's beneficial. Yeah.
Yeah,
Uh-huh.
and through some
Okay.
random process, these
And
de novo genes have to randomly form that
uh-huh.
make the useful proteins.
You
That's
know
the
we've
mechanism I'm
you know
saying
we've
doesn't
directly
exist.
observed this, right?
Name it.
Name it.
Name the time
Okay.
it's
Arctic ice fish. In rice. In humans, we've got de novo genes. And here's the thing.
Arctic
You, as
whitefish.
a creationist, accept these examples because if you do not accept that de novo genes evolve, then you are going to posit species level creation. Now, are you a species level creationist?
Let me
No,
point out
answer that
the
question.
fish.
Are you a species level creationist? Is it your position that each species was created independently?
I believe in adaptation by mutation and loss of information.
Okay, so that's not an answer to the question I just asked, to be clear. Do you think that at creation there were like the ancestors of kinds that subsequently became different species after the flood? Or do you think that each individual species was separately created?
I'm going to point to the
No, you are going to answer the question because this is an important question about the argument you are making. And it has implications for whether or not de novo genes have evolved and whether we agree on that. So I want to know about your model of creation. Is it at the species level or the kind level, whatever you want to call that?
I'm saying it's at the kind level, and I'm going
Okay,
to back
great.
up. I'm going to back up
So
my
we agree,
statement, what
no,
I'm
no.
saying.
Now we
with a study.
agree. We then agree that de novo genes evolve because you accept that kinds were created and speciation happened after that. So first of all, you agree that macroevolution happens. That's great. But
That's not true.
specific speciation is macroevolution. If you believe that kinds were created and speciation occurred after the flood, then you accept that macroevolution happens. But more importantly for what we're talking about right now is that if you accept that you have kinds that were created and then speciation happened, you have genes that are unique to each of those species that were 100% not present in the ancestor because that whole region of the genome is missing in all of the other members, which means we agree That
we
de
don't.
novo gene origination. So where did that extra stuff come from? Was it created to look like it evolved, even though it was actually created in place? You just said kinds were evolved, not individual species. So those sequences had to appear at some point in their history.
So 2018 mitochondrial barcode study.
Nope. I'm sorry.
I-
You're gonna
you-
do.
you
You're
want-
gonna bring up Stokel and Thayer here. Okay, fine.
Yeah.
Bring it. Let's.
This
First
is-
of all, I
this
just
is-
hang
I'm
on.
just-
I'm gonna talk for a second, and then I'll let you do whatever you want with the barcode study. I just want to point out for everybody that that whole thing about de novo genes and how they got there. Let's call them. You could say orphan genes or taxonomically restricted genes, right? How they got there is a major problem for every creation model, because either you accept species level creation, in which case those sequences didn't have to evolve, or you accept kind level creation, but then those sequences had to come from somewhere, and either they evolved or God put them there, and then we're doing last Thursdayism. It's completely unfalsifiable. So that's the point that Glenn just ran away from there. Now we can talk about this silly mitochondrial barcode paper.
You're putting definitions that I don't believe.
I don't care if you believe them, because those are the definitions, my guy. Like,
Your definitions. Speciation is just a mutation that creates a slightly different feature. That's it. Just like dog breeds.
so, and that's macroevolution, because speciation is macroevolution.
That's micro, because all it is
Nope.
is a change in an allele that creates a different, birth defect. And then
That's
the normal
not
allele is lost, and the change is permanent.
That's not speciation, though.
See,
You
your
need to have
definition.
you need to have a divergence in order for speciation to happen. What you just described isn't speciation.
Darwin's finches.
That they had divergence, which is not what you just described.
Yeah,
Right.
and so they have new genes. Is that what you're saying?
I don't actually know. I don't think any of them have any de novo genes. It's a change in expression of genes during development. I forget the name of the genes, but it's the gene that dictates the beak length and depth. There are two genes.
It's the ALX1, and it was a point
Oh, great.
mutation. It was a point mutation
Great.
that made the beaks change.
Made it bigger. Yeah. Uh-huh.
Yeah, that's it. A point
See, now
mutation.
we're just going in circles, though, because this is where we started. So why don't you go to
No,
that
you're-
mitochondrial paper?
Every time I get to the point you don't want to answer, you start interrupting.
I don't know
I'm
what your
saying-
point is.
My point is that's microevolution. That is a mutation
I
that made
agree.
a change. That made a change in a
I
beak.
agree.
Microevolution,
Yeah, because that's
And
not
it created-
speciation. Exactly.
and it created a new species.
No, it didn't. But I mean, you're wrong. The
Oh, it didn't.
the new species that was recently documented recently within the last few decades in the Galapagos Islands was a hybrid. It wasn't a divergence event. It was a hybridization event that led to a new population that was reproductively isolated from both parents. That's different from what you're describing. So,
What I'm describing is in textbooks. They
okay.
have pictures. This
Which
beak,
ones?
that beak, and that's why they call them a new species, Darwin's finches. Otherwise, they'd have just been finches with thicker beaks.
You are
Darwin's
okay.
finches
Do you
are a
realize
new
Darwin's
species.
finches isn't a species? Do you think Darwin's finches is a species?
It's an example of a change.
No, but no. Answer that question. Do you think Darwin's finches is an example of a species of finch?
Oh, I don't think so. That's just what it's called.
Okay. How many? Okay.
And I'm
If
giving
somebody
you the
was
mechanism.
to ask, let me put it this way. If somebody was to ask a bird ecologist, how many species of Darwin's finches are there? What do you think they would say?
I have no clue why you're
Okay,
even
then maybe
going,
you should
going
read a
you're
book.
changing, you're going off on a tangent.
No, because you keep talking
I'm
about.
talking about
It hit me just
mechanisms.
now.
I'm
It hit
talking
me
about
just
a
now.
mechanism. A mechanism.
You
how it
think? Hang on,
happens.
no, wait, just hold on one second, because I think you think that Darwin's finches is one species, and then after the drought, they became a different species. Is that your impression of what's going on here?
No, I'm saying there was a beak that was normal. A drought happened. There was a mutation on the ALX1 gene, point mutation, made
We heard
thicker
this
beaks.
already. Yeah.
Yes, and because of the drought, they were thicker nuts that the beaks that were thicker could crack, and the food source was better.
How?
Therefore, that population survived better.
I'm
So
gonna
they lost the allele for the small beak, so the population changed to having all thick beaks.
and
And people called them
that
a
a
new
new
species.
okay.
That I didn't.
No. No.
That's exactly what's in the
I'm gonna get to
textbooks.
the point here, folks, because I think what's going on is Glenn here thinks that there's one species of Darwin's finch that experienced a speciation event with the drought in the night. Tell me I'm wrong. Then you tell me how many species you think there are. Because as far
I'm
as I
not
can tell, you're
talking.
describing it as one species.
I'm not
There are
talking
actually like
about.
over a dozen. There are over a dozen species of Darwin's finches on the Galapagos Islands, and Darwin's finches is a collective to describe all of them. It's not describing any specific species of finch.
See, I'm just talking about the mechanism. I don't care how many finches there are. I'm talking
Well, it
about the
matters because,
specific mechanism that made the change was a point mutation,
but
and then it changed it, loss of an allele, the population stayed that way.
do you
That's the
understand?
adaptation. That's the adaptation and the evolution we see.
Do you
That
understand?
mechanism.
Do you understand that what you just described is not speciation? Do you understand that? That's not a question
I didn't.
of
I
is it
didn't
or isn't
say.
it. What you
I
just
didn't
described
say. I
is
didn't
not
say it was.
speciation.
I didn't say it was. I said
Okay.
it's described in the textbooks that way.
No, it's not.
Not
And
me.
I'm telling you that it's not. And I'm telling you this based on the fact that I teach this stuff multiple times per year. So in the textbooks I use, the thing you just described is not a speciation event. You are confusing the change in beak size in the drought in the 70s with the initial adaptive radiation when the islands were first colonized by the finches from the mainland. Like, you
Okay.
are conflating those two things.
So.
That is
So
what you are
all
doing.
the books, the textbooks, the diagrams that have that exact same thing are wrong, and you're right. Got it.
Thank you.
Okay. But we still got to get back to the point where
Which is
you
macroevolution.
you can't answer the question of how the millions of proteins could be created by some
I
mechanism.
gave you 5 mechanisms
You
of how you
didn't.
get new proteins.
Gene transfer doesn't create new information; it transfers. There's no new
That's
genes.
new. That's new. You gain a new gene. That is new. You gain something that wasn't there before. That is new. Now you have new stuff. Even if it's the same, here's the thing that a lot of people miss. Even if it's the same protein, the fact that you have another copy of it means you might have a different phenotype. It means you're going to have more biochemical interactions happening. It means you can have a mutation with no cost that leads to different biochemical interactions. So yes, horizontal gene transfer, gene duplication, mutation, de novo gene origination. If we're talking all the way back, we're going to talk about bacteria. We got to talk about plasmids, right?
Okay, give me one example, observed example of a new gene creating a new protein, useful protein.
The ice fish antifreeze protein.
See, when I see the ice fish, I can easily say you had a fish that's in a population where the water was extremely cold. We had a change in the weather where it was warm, where the antifreeze in their system was detrimental in the warmer water. So they lost the gene to make the antifreeze. And you can't say that didn't happen.
Yes, I can, unless you want to go with species level creation, because we can compare not just one fish to another fish, we can look at collectively all the fish. And if you're saying that gene if you're going to say that gene existed in the past, then what you are proposing is that every other group of fish in that created kind independently lost that gene in exactly the same way with no trace. No pseudogenes, no deletions at different places in the genome, no remnants of that gene left behind. You're saying that dozens, if not hundreds, of species of fish had to experience the exact same nucleotide perfect deletion. in their genome without a trace. So that gene existed in the ancestor, and it's still there in the Arctic fish, but all the non-Arctic fish lost it in exactly the same way. That's what you're saying is more likely than a new gene evolving. Those are the lengths you're willing to go to deny that a new gene evolved there.
So you're telling me that it's beneficial to have that gene in warm water?
No,
No,
I didn't say that at
right?
all. Are those the words I said? Chat,
No.
are those the words
Okay,
I said?
let me start over. You
No,
got a you got a
please
particular,
start over and try not to
you got a
misrepresent what I
particular
said this
you
time.
got a particular fish with antifreeze, right?
Yes.
What fish was it before that it came from that developed the antifreeze?
I don't know my fish phylogenetics. It was a
Exactly.
non-Arctic
You
fish.
don't know. You don't know if that's the only one that ever had it.
It's the only one that has that particular antifreeze gene. There are
Exactly.
other antifreeze genes because
Exactly.
Antarctic
So
ones
you-
also
so you
have
can't
antifreeze,
say-
but it's a different gene.
So you can't say it wasn't created that way.
I can say it wasn't created that way unless you want to propose species level creation, or you want to propose ongoing divine intervention to make new genes when necessary. If you want to propose either of those two unfalsifiable things, then I cannot say it wasn't created that way. But since you say that kinds were created, not individual species, then I can say it wasn't created that way. Because again, it would require every other member of the kind to have lost the gene in exactly the same way with no trace. And we know what it looks like when you lose a gene. That's what pseudogenes are. We know what deletions look like because they often leave behind remnants of the region that gets deleted. So we can see like, oh, there's this little tail of a gene here, but in this other species, you've got the whole gene. So it looks like most of it was deleted in the first species, right? We know what that looks like. So you're proposing that that gene got somehow excised out of other species, and we have no trace of it. It happened multiple times independently in identical ways.
And you have no example of
What's the
where it came
probability of
from.
that?
You
What's
don't
the
know where it came
probability
from.
of that?
You don't
We
know where
do.
it came from.
We do know where it came from because it's mostly repeats. It's a series of duplications.
What fish?
The Arctic, I forget the name of the fish, Glenn. It's the Arctic fish with the de novo antifreeze protein. We all know what we're
Yeah,
talking about
but-
here. That
but-
protein is largely a series of repeats of the same amino acid sequence.
I don't want to talk. Here's
And,
the
well, hang on. No, you asked where it came from. I'm giving you the answer where it came from. It's a series of duplications that lead to this protein that is largely repeats, and then it acquired what's called translational competence, which is when you get a start codon and a stop codon. That's the answer to your question of, you say, I don't know where it came from. No, I can give you a blow by blow of where it came from. We know where it came from.
I've read the paper. It's just an assumption.
Okay?
There's no observable evidence. It's just an assumption. I've read the paper.
That's an assumption. Okay.
Yeah.
You want me to give you some more observations of macroevolution? So we did de novo genes, we did speciation. You want to talk about? I know you love the multicellularity example. You want to talk about
Well,
Chlamydomonas real fast?
how about we go from dividing a single cell to meiosis?
Meiosis.
Yeah, meiosis.
Okay. What's the pro?
How did that happen?
How did that happen?
Yeah.
How do you divide chromosomes?
How did you go from a single cell just dividing by either mitosis or just budding or whatever,
Right.
to sexual reproduction or meiosis where it's dividing, whatever? How do you go from single cell
I'll
to
tell you.
multicellular?
Oh, it's so well. That's a separate question. Single cell
It's
and
the same thing.
multicell
This
is
is
a
what I'm
different
getting at.
question.
What I'm getting at is,
So which one do you want to do? So I'm happy to do either.
how do you go from a type of division for a single cell organism to one that's a multi-cell?
They do mitosis and then they stick together afterwards, so you form a clonal cluster. That's how.
No, you're not talking. How do they divide?
What do you mean? How do they divide? They do mitosis.
How do they multiply?
The word was in the answer I gave you. So you have a
They
single
didn't-
cell, right? It does mitosis, and when you do mitosis, you have to do two things. You have to separate the nuclear material, right? And then you have to do cytokinesis. You have to separate out. Now, once you do cytokinesis, you have to actually separate from each other once there's plasma membrane separating the two separate cytoplasms. Yes?
Right.
With me? Great. Sometimes when that happens, the cells stick together. That can be beneficial in some cases. So you actually have selection for the cells that tend to stick together instead of completely separate.
Didn't you have to apologize for this?
Nope.
Because this is not multicellular. This is just a single cell organism forming a protein mat at a colony.
Wrong.
And it never changed the way it
No, that's wrong.
replicated.
No, that's wrong. This is again a very common creationist talking point. You see,
I
hear
read the
this
paper.
from no, hey, you hear this one from
In
Dr.
the
J.
conclusion,
Weil. You hear this one from is the one that I know of. But other creationists say this as well. That the green algae in question, the unicellular green algae, the Chlamydomonas, who never have any multicellularity in their evolutionary history, they can facultatively form colonies by having individual cells stick together. That's a response to predation. That's separate. That is a separate phenotype from the de novo multicellularity explained in that paper, the Heron, I forget what year, 2018 I think it was. That's a different thing because the proteins that are involved in the extracellular matrix in those two phenotypes are different from each other. So the de novo multicellularity in that paper is not the same as just permanently doing the clustering thing. Instead, those cells, they divide, but they stay together, and they do that multiple times to get 8 or 16 cell clusters that are too big for the predators to eat. That's the optimal size, right? That is a de novo form of multicellularity in an organism that never had multicellularity in its evolutionary history. It had facultative colonialism, which is different because that's individual cells that were already separate and not directly from mitosis with each other coming together. Okay? You are getting your information from creationist sources that are misrepresenting the studies to you.
I have read the paper. I know exactly. The cells just didn't completely divide apart from each other. They just kept one membrane around them. There's still two individual cells.
They were up to 16 individual cells.
Yeah,
and I actually think they got up to 32.
16 individual cells inside of a membrane still replicating as an individual.
For folks in the chat, the paper is it's Heron. I'll put the name in the chat. It's Heron et al. De novo multicellularity. If you search that, you'll find it. There. It's Heron et al. De novo multicellularity. You'll find it. That's the paper. And so I want to be clear about this because what you just said is what I just explained how it's wrong, okay? What you just said is that when they separate, they don't completely separate, they stick together, and that's not new, right? That's just doing whatever old thing they could do. That's wrong. The extracellular matrix that they use to stay together in the de novo permanent multicellular lineages, the ones that stayed multicellular for 2 years even after you removed the predator, so it wasn't facultative, that extracellular matrix was unique compared to the ancestral state that they used to stick together.
Okay.
Like, I want to be clear about that. You are parroting what you have been told by creationists, and they have lied to you.
I read the paper.
Then you have misunderstood the paper. I was giving you the benefit of the doubt.
16 individual cells replicating as individual cells inside of a cluster.
No, that's wrong too. They didn't replicate as individual cells. They had mating structures called propagules. So only a subset of the cells in each group were replicating, which is a hallmark of multicellularity. They were clonal, individuals. And then only a subset actually propagated the next individual.
So you're saying 16 cells copied themselves and became a brand new set of 16 cells separately.
You have 16 cells, and then they would spin off 4 reproductive cells, which would then clonally grow into a new multicellular organism.
Yes. Individual cells in a colony.
Not a colony. Not a colony.
Inside of a membrane that's a protective membrane.
It's an extracellular matrix.
Exactly.
Which is not a membrane. Which is not a membrane. No,
But
membrane is
it's
made
an
of lipids.
extracellular,
Extracellular
whatever,
matrix
but
is made
they're
of
still
proteins. Those are
inside of
two
it.
different things. Membranes are lipids. Extracellular matrix is proteins. They're two different things.
Okay. So the process of making a new cell,
Mm-hmm.
right? How did that change?
What do you
The
mean? How
actual
did it
process.
change?
How do you get to meiosis?
So you want to change the topic from multicellularity to meiosis now?
That's what I said. Single cell type replication, like mitosis, going to meiosis. How do you do that gradually with evolution?
How do you gradually evolve meiosis? So you're putting the cart before the horse, because what you have to understand is that prior to sexual reproduction, you have recombination that's decoupled from reproductive processes. Do you understand that? That you have recombination separate from reproduction prior to sex evolving. Do you
But
understand that?
there's a process.
No, please, do you
Meiosis.
understand what I just said? That
You're
recombination
avoiding my
predates the
question.
evolution of sex. Do you
You're-
understand that?
Where did it come from?
Where did it come from? Recombination is as old as the oldest cells.
Recombination does not create meiosis. It's a
Right.
completely
It
different
Right.
process.
It predates meiosis. I'm asking if you understand that.
Yes.
Okay, good. Great. Good. Good. Now, the thing with meiosis is you go from a diploid cell to a haploid cell. Right? You know what those words mean? Diploid is when you have two copies of everything. Haploid is when you have one copy of everything in your genome. Now, with recombination, among prokaryotes, what you can do is give your entire chromosome to a different cell. And now that cell is going to be diploid, right? Because now it's got two of everything. Yes?
Yeah.
And meiosis is the process of taking the diploid cell and returning it back to haploid. So when that cell divides subsequently, each daughter cell gets one of the complete sets. Ta-da, you've done meiosis.
That's not meiosis. Not true meiosis.
Crossing over already existed. Because when you do recombination, whether you're talking about prokaryotes or eukaryotes, when you do recombination separate from reproduction,
crossover is not
when-
meiosis that we see
correct.
today.
Crossing
that we see
over is
today.
not meiosis, but it's
You
an
still-
important
you-
component of meiosis.
You still
and
have to
it is
get
the
there.
basis for it. Because the whole point is you're haploid. You take somebody else's genes. Now you're diploid. You recombine, and then you divide back to haploid. And now all those offspring, they're not clones. Now they're all different from each other because you did the recombination on the front end. That's meiosis. It's actually, technically I described fertilization and then meiosis, but it's the dividing part to go back to haploid that's meiosis. That's how it happens. It starts with recombination that predates it.
It's not what's done today in multicellular
What I just described
organisms.
is literally what's done in paramecium.
In multicellular organisms, meiosis, sexual replication, whatever, it's different than that.
Right, because I just described, you wanted to know how it evolved, so I told you how it evolved in unicellular organisms, because that's how they did it first.
Right. So the evolving process, okay, it's going to happen slowly, right?
And not necessarily. Some evolution happens fast.
Okay, but it can't happen all at once, like boom.
Sometimes it can
go in steps.
Sometimes it can.
Not
Like
enough.
whole genome duplications are very common, and lots of organisms are very tolerant of them. Multicellular organisms, yes.
We're talking about a change in the way a cell divides. That can't happen instantaneously differently. I think you would agree with that.
A change in the way, but I didn't describe a different way cells divide. You do recombination first, and then you allocate your chromosomes. It's very similar to mitosis. You just start with an extra set, and then you don't do DNA synthesis. You don't do replication. So you reduce the number of chromosomes over the course of the division. Like, I don't know what the disconnect is.
So you're saying that we that's how we reproduce, like that.
we do do you want me to have the talk with you? Like we do
Meiosis.
meiosis in the
Meiosis.
germline that makes egg and sperm, and when a man and woman love each other very much.
And that's not what you're talking about. You're
You
talking
asked me
about
how that process evolves, and I'm giving you the answer.
it has to evolve in steps. And once it
Yeah,
goes
and I gave you the first step.
once it goes into a step that's a little bit different, then it's non-functional.
That's not true. That's not true at all. There's lots of
Oh,
variation
so
in this process. So, for example, the
okay.
reproductive cells that result are called gametes, and you have differences in the size of gametes, right? Some gametes are bigger than others just because some things make cells that are bigger or smaller than others. Once you have gametes that start exhibiting polymorphism in their size, now game theory takes over, and you typically end up with two types of differentiated gametes: very large eggs and very small sperm, because those are the combinations that are optimal for each of those two strategies, and you lose having the undifferentiated gametes that are intermediate in size.
Yeah. So
Right?
you get to sperm and eggs,
I don't know.
you
A
know.
minute ago, you were complaining about me explaining the origins of the process, and you wanted me to explain how we do it. So now I explain
I know,
where
but
egg and sperm come from.
you're gapping to sperm and eggs, and then you've got a process to use them. Which came first?
Oh, now we're doing the okay. So you need to have a male and a female of the species. Blah 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 blah. Yeah. Again, sex doesn't work that way. The
I
cells and the recombination come first. The specialized morphologies come later. Like I don't, I don't know, man. I
see your process of just a cell picking up another nucleus or whatever, and then dividing that
that's
out
yeah. That's literally
is
what
not
fertilization is.
it's not fertilization like
That's
us.
literally what fertilization is. You have one cell, and that cell gains another nucleus. Ta-da! That's fertilization.
Okay,
That's the
so
definition
that's
of the word.
so that process is exactly the way we do it. Is that what you're saying?
We do fertilization. Yes, that's how we do it.
And so the your example with the bacteria is the same process.
No,
Exactly.
bacteria do recombination differently because they don't have nuclei. So they do
Exactly.
recombination differently, and it's decoupled from reproduction.
And so that process would have to gradually change
And
to
how do you
our
get to it? And I
process.
explained to you, once you have a nucleus, when you do your recombination, that leads to you going from haploid to diploid. Once you're
And
diploid, right? Because
that,
when you gain a whole other set of stuff, now you're
yep.
diploid, right? Because that's what that word means. Now,
Mhm.
once you're diploid, you do your recombination, and the chromosomes line up biochemically because they have an affinity for each other because they match. So they line up and they do their switcheroo. That's an old process. We don't have to evolve crossing over because that predates all of this stuff. Once you do that, then that cell divides, but it skips a round of DNA synthesis, so you end up losing half the genetic material. You go back to haploid. That's meiosis. Like you're
And
making this
so
seem like it's a completely new thing,
no,
and it's
here's
not.
my point. Is is all of this new way requires new proteins to pull the cell to pull the DNA
No, that's not
apart to
true at
make
all. What new name them?
so
Name them? What new proteins do you need for
so
meiosis that you
the
don't
so the
need for mitosis?
proteins in
Name them.
mitosis
Name
the
them.
proteins on mitosis that pull apart the nucleus are the same proteins that we have in ours for meiosis. Is that what
Yes.
you're saying?
Yes.
They're exactly the same.
Yeah, it's the kinetochores, and the enzyme is called separase. Yes.
And they're the same. Same
Yes.
as us in bacteria.
No, not in bacteria because we're eukaryotes. I don't know why this is hard. Like, it's a bacteria do it differently, as I've said.
Exactly. They use different proteins
Yes,
for
and
their
we went through again.
replication.
Okay, let's do this again. Gene duplication, mutation, horizontal gene transfer, de novo gene, or right? We've been through
So
this
you
before.
need
We have
you need
lots
the
of mechanisms of
proteins.
getting new proteins.
You need the proteins first to make the system work.
That's not true.
How do you? So it's going to work without the proteins.
It's going to work using the ancestral proteins, and then they specialize later on. Yes.
So they just randomly just find a whole new protein that says,
No,
this one's going to work better.
no. You
So you're just
have
making shit up.
uh,
You
Okay.
have no evidence for any of this.
I have no evidence for any of this, except I've got like empirically validated phylogenetic methods. I've got direct
there is not a
experimental
single paper.
observations. I've got
There is
direct
not a
experimental
single.
observations.
There's not a single paper published that says that it explains how you can go from mitosis to meiosis.
There's not a
It's
single
just a
paper
guess.
published.
No.
Yeah. You want to do something fun? Share your screen and Google that. Prove it.
What?
Share. Go on Google Scholar. Share your screen. Go on Google Scholar, and Google. Go on Google Scholar and Google evolution of meiosis. Go. Try. Let's do it. Let's do the experiment. You're saying there's not a single paper that explains how meiosis evolves. Have you ever? Do you know how to do Google Scholar?
No.
No,
This is the first
this
time.
is
I don't know.
amazing. This is you're sitting here telling
I don't
me
know how
that
to- I
you
don't know how to
understand
share
this
a
stuff
screen.
better than I do, and you have never used Google Scholar. This is
I don't-
incredible.
I don't know how to put it on a screen here.
You go
I've
to the
never done
bottom
this
and hit
before.
share screen.
That's what I'm saying.
Yeah, you go to the bottom on StreamYard, and there's a share screen. It's the monitor with the plus. I can't wait to hear how all of the papers that you've never read don't count once you pull up the screen. I can't wait. In fact, I've got my textbooks right over here. Let's see. Where's my? Hang on. I got to take this off for a second here. Oh, actually, I could just put it. I could just Google it right on my screen. Hang on. There we go. Nope, that's okay. So you're sharing your screen, but it's the StreamYard window. You got to go to the other tab. So you want to go to the Google. You have your browser.
How about you pull the paper
in
up?
your no, because I want you to do it.
I don't know how to get navigate.
You don't know how to
No
do
one
this.
wants
Is
to-
I can we just appreciate how rich this is? That you're here telling me that I'm wrong about everything I'm saying, and you don't know how to use Google Scholar to look up the literature. Can we just appreciate that for a minute together?
I don't know how to use this system to pull anything up. Is
So
what
you
I'm
have your
saying.
screen pulled up, but I'm seeing it's the StreamYard window in the preview.
I have no
All you do
clue.
have to do is switch to a different tab. There you go. That looks like is that an email? That's an email inbox. You don't want to show that.
Okay. All right.
Everybody, I apologize that we're having this little, downtime here, but I think this is a useful exercise. There we go. That looks right. I see Google at least. You're going to add that to the stage. Good. Now you're in Google Scholar. Now put in evolution of meiosis. Because you're saying there's not a single paper on this.
Right.
The
Let me
first one
see.
looks informative.
The
do you think?
origins of meiosis in early eukaryotic history has never been satisfactorily explained.
Okay, so you're doing the thing where you quote the first line of the abstract.
No,
where
I'm
the
saying,
authors
is this the
set
one
up
you,
the
is this
problem,
the one you want?
and then they
Is this
solve
the one
it.
you, I'm just
Yeah.
reading the first line. Is that the
I
one
know,
you
and the
want?
first line, so this shows me that you've never read a scientific paper past the abstract, because what the authors do with the first part of the abstract is they set up the
Come on,
problem,
Dan.
and then they
I just-
explain
the
how they
only-
solve the problem.
I know this. The only reason I read it is I just read the first sentence and asked, is that the one you wanted? There's a bunch
You
of
can
them
click
here.
on any of them. I'm just want to show you, because you said a few minutes ago that there's not one paper that explains the evolution of meiosis, and I don't know, man. What do we think? You let me know when you want to wrap up this little exercise here. As somebody in the chat clocked, I'm having way too much fun with this, so I'll let you, Oh, I'll let you experience this as long as
I
you
don't
want.
know. I don't see anything in here that says anything about them.
Maybe you should read past the abstract sometime.
Well, that's all it goes to.
Good thing there's more than one. All right. It is getting late. We have moved far
My point
afield.
is that you've never answered the question of what's the mechanism other than saying gene transfer, something like that.
Oh,
You
I
can't,
never
you
answered the
can't.
question of what's the mechanism except for giving you multiple mechanisms that we've directly observed.
So you've got to do something like a single cell, add a body part, or a function. How do you get those proteins?
I gave you answers
No,
to those
be
questions.
specific. Not just name a thing.
Yeah, so the fish
Do
antifreeze
specifically
protein.
the mechanism within that thing.
Yeah, so the fish
That's what I'm
antifreeze
saying.
protein was a duplication of a region multiple times that led to the de novo protein, plus single point mutations that led to translational competence. The HIV novel function that involved 3 new binding sites
We're not talking
was
about
a series of point
HIV because
mutations.
that's an existing structure. It
It's a
just
totally
mutated.
new biochemical function, 3 new binding sites, totally new
It's got a
biochemical.
four amino acid substitution. It's
Up
not
to 7. It
something
was
new.
only well, it was 2 regions. One of those had 4. The other one they didn't count. So it was up. It was at least 4.
Yeah, an existing virus.
Yeah, an
Four,
existing
four,
right.
whatever.
Well, it's a new species of virus. To be clear, it's a new species of a virus.
Okay.
HIV was a new species.
But it's still a virus.
Oh, now we're doing it's still a virus. Great. Now
No,
we get
we're
to talk about photosynthetic
you
animals.
see, you're skipping.
Have
You're
you ever
trying
heard of
to
the
avoid.
photosynthetic animals?
You're trying to avoid dealing with the mechanism. You cannot come up with a mechanism that finds the correct proteins, and you can't find a paper that
All
says
right.
it.
All right, we've already had this conversation 3 times tonight, so we're not going to
No,
do that
you've
again.
never
If
answered
anybody
it.
wants to hear the mechanisms, you could just go back to one of the other 3 times we did it. But I want to just end with
You
this, 'cause
you
we
you
will wrap
listed
up soon.
four generic things, and you never explained how they could do it.
I wanna end with this because you just said it's still
See,
a
now
virus.
you're running, just like with
No,
Hovind.
I already gave you the answer to your question
No,
multiple
you did
times.
not.
So
This is just like Hovind.
mutations,
Okay, mutations.
gene duplication,
How-
horizontal gene transfer, de novo gene origin. Like I can give you like those are mechanisms. You don't have to like that those are mechanisms, but those are mechanisms that we have directly observed. So now that I've answered your question a 4th time, now I want to end this by asking you, since you did
You
still
have
just
never.
a virus.
You have not given an example of how.
I gave examples of all of those things. I'm going to ask you this question. You don't have to answer it if you don't want to. I'm going to ask you the question. This is the topic we'll end on because we've been going for a while. Are you familiar with the animals that are becoming photosynthetic? Simple question.
No, I've not heard of that.
Oh, okay. Well, that's a really fun example because this is a case where you've got animals that eat algae and they steal the plastids, the photosynthetic organelles from their algal food. It's a process called kleptoplasty. Dozens of animals do this, and they use the photosynthetic organelles.
You've
Now,
got
at
two
least
things.
one
Two things
species-
created already, and
At least one species of these animals has experienced horizontal gene transfer from the organelle into the nuclear DNA of the animal. So you've got the animal that's expressing genes for photosynthesis, right? So this is a clear case of animals becoming photosynthetic. Now you just said, I bring this up because you just said it's still just a virus. Is a photosynthetic animal a new kind?
no, it has
No.
no feature.
A
It has a new feature.
photosynthetic animal is not a new kind. That doesn't count as a new feature. That's
Is it-
amazing.
Is it a bacteria?
No, I told you it's
What
green
is it?
algae. It's a green algae.
Okay,
It gets
it's
eaten
green algae.
by
Now
hydra,
what is
sea
it?
slug. There's a bunch.
Is it still green algae?
No, the green algae has gotten eaten, and it's becomes an organelle in the sea slug, which is now photosynthetic. So it's an animal, but it's photosynthetic.
So the sea slug is still the sea slug.
It's now photosynthetic. Yes, it is a
Yeah.
new type of sea
So
slug.
it has just a new feature.
Yes, like you've been asking
Yeah.
for.
Yeah. So it's never going to change into something else.
So it's your contention. So this is a great place to end it. It's your contention that animals
You,
becoming photosynthetic is not macroevolution?
you still have
No, no,
never.
just answer that question for me. Is an animal becoming photosynthetic macroevolution?
Where did the genes come from?
Yes or no? It's a simple question.
No.
Amazing. Amazing. So here's what I think we could take away. That's perfect. So an animal becoming a completely different type of organism metabolically
A
is
gene,
not
just a gene
an
inserted.
example. No, no, hang on, let me get this out. It's not an example of macro. That's just microevolution. Is an animal becoming photosynthetic? That is perfect because that is exactly where this always ends, where every observed example of evolution is microevolution. And I'm gonna end this, and I'll give you a chance. I have one more question for you, and then we can wrap this up. I'm just gonna end this with something I read on Reddit years ago, and it's I apologize to whoever said this initially. I forget who it was, but they said microevolution is evolution that creationists can't deny. Macroevolution is evolution that creationists must deny. And what we're seeing here is that Glenn must deny macroevolution no matter what it is, no matter what we observe. So I have one more question before we wrap this up, Glenn. Going back to something I mentioned earlier. You said 10 minutes to embarrass me. How'd that go?
You never let me ask the questions. You kept interrupting. You always wanted to argue about definitions.
Yep.
To change the subject, and you never answered the question, how does it specifically happen?
Okay.
You just gave 4 categories, but you never went into detail. How can it find the correct proteins out of an infinite number of possibilities? And you just keep running from that.
I just want to point out that I asked how the Operation Embarrass Dr. Dan was going, and you didn't answer the question.
I just
How
did
you
it.
feeling?
I just did it.
Can we get like
You can't answer the
post
question.
game?
I just did it. You can't answer the question.
All right, I answered about 4 times.
No, you
All
didn't.
right.
You gave 4 categories and you couldn't answer the question.
All right, folks. This has been
exactly what
something.
you did with Hovan.
This has been something,
When
folks.
it got
He's
to
still talking. We're trying to wrap up the stream, and the man can't help himself. He's still talking. Okay. We're going to wrap this up, folks. I hope you all had as much fun with that as I did. Because as some of you in the chat clocked, I definitely had way too much fun with that. So thank you all for being here. I appreciate all of you. Hope you're all healthy. Have a great night, and I will see you. I'll see you down the road. I got a video pre-recorded. I recorded today for next week, and then I think I'm going to do a live show the week after that. So keep an eye out for stuff. Thanks, everybody. Remember to hit the buttons before you go. Leave comments, all the good stuff. Have a great night, everyone.